Good evening, everyone. It's a real pleasure and a true privilege to be standing in front of you tonight. I hope you can hear me well. I also like to take this occasion to thank Spy for giving me this opportunity to talk to you. So I'm working on trying to understand why some of our genes are expressed in a very consistent and predictable manner, while silver, on the contrary, are expressed in a very variable and fluctuating fashion. And for that, I apply complex statistical analysis on large-scale data sets. But this is not the story I'm going to tell you about at all. Today, I'm going to tell you about something I call the changing face of biology. And in a sense, it's just encompassing two major ideas. The first idea is that the biology of our cells is extraordinarily complex. But this is truly important to try to characterize that complexity. And this brings me to the second idea of the talk, which is that we're perhaps standing at a stage where technology is enabling us to actually tackle that complexity. So it is truly an exciting time to have a better understanding of the most challenging diseases that we're facing, and also for biological applications. But without any further ado, let's embark on that journey, which starts with the DNA molecule. You've seen wobbling for long enough by now, because the DNA molecule carries, as you know, our genome. In a matter of fact, it's actually only 2% of our DNA that codes for what's called proteins. And really a central question of molecular biology and biological sciences as a whole is how, when, and what for protein coding genes are expressed. And so there is something quite useful in biology called the central dogma that was developed from the late 50s and onwards, and that in essence tells us that the genetic information flows from DNA into proteins in a way that you first transcribe some messages on the DNA here in red into something called a messenger RNA. And that messenger RNA is processed and transported into another part of the cell where a big machinery called the ribosome translates that message into chains of amino acids here in red, which are the building bricks of the proteins. So proteins adopt a three-dimensional structure, and in doing so, they adopt some functionality inside the cell. And really, any perturbation in that system can bring deleterious consequences for our cells. Right? Typically, those perturbations can take the shape of mutations. So it means that if you change any sequence in the DNA, it can affect, first of all, how the gene is transcribed. It may also affect how the messenger RNA is translated. It may affect the way the protein folds or functions. And quite luckily, since probably 2000, and in the advent of the human genome sequencing, we've been lucky enough to actually be able to sequence most of our genes, DNA, RNA, and protein sequences. So this is really truly something amazing, and it's brought a lot of promises as to how we would be able to tackle disease by knowing the sequence of our genes. Right? And another key question that I mentioned earlier is the what for proteins are expressed. So trying to uncover the functions of proteins. And for this, you really have two different scales at which you can work. One is really molecular, down at the molecular level. So you try to really characterize what a specific individual protein is doing, how it associates with other proteins to form machineries, and what those machineries actually do, like performing tasks like converting nutrients into energy, transporting things, replicating, and so forth and so on. And there's also a more broader, holistic system-level view where you don't try to focus only on individual proteins, but try to see how, as a whole, they form a protein-protein interaction network. So a sort of giant entity that permanently and dynamically rewires and reshapes in response to the environment. So these two views are really particularly important to try to tackle a lot of complex diseases, as we'll see. Right, but the problem here is there's a lot of caveats in that picture. It sounds quite simple, but as a matter of fact, if we revisit the central dogma in the light of what we've discovered in the past 10 years or so, there's a lot of events that happen during the central dogma that really complexify and sort of blur the relationship between unique genes and unique proteins. In fact, it's a one-to-many situation. So I won't go into the detail of all these different phenomena, but the global idea is that they really create a gap between what we know from the sequence and what we observe in vivo, meaning that a cell in a given tissue in your body might have a whole diversity of different protein versions that have been synthesized from the same gene. So if you want to understand the impact of mutation, you need to understand that as well. And we actually have been struggling a lot. And I think in some sense, one could argue that it's also slowing down a bit of the innovation, because we're facing an avalanche of complexification, if you will, or like a lot of new data sets that bring new light, shed new light on how this process works. So we're actually failing at it somehow. So the real question to ask first is, why do we fail at handling such complexity in the first place? And there will be many reasons to answer that question, but I could really name and nail down to 3 of them that are really fundamental, I think. The first one is we're actually, when you're studying sub-networks of proteins, so let's assume like we have a protein complex made of 5 partners that are regulated by 2 other proteins, right? We're, in general, bringing a lot of knowledge from many, many different papers and many years of studies, but we actually don't know what's happening dynamically. Because if you look behind the nodes, the expression levels of the components of the system might vary, and it will vary in function of all the events I've described of transcription and translation and also degradation, because all those rates of transcription and degradation can be tuned thanks to regulation as well. So it's really a complex picture, something that evolves over time that we need to characterize. So that's the first problem, and it's all about regulation. The second problem is a problem of multidimensionality. Biological systems do not stay in one given context. Actually, the context changes all the time. So you might imagine, especially if you're studying mathematics or something like that, that you have an object that really flows into multiple dimensions. What do I mean by that more specifically? Well, first of all, cells do often change the environment they're exposed in, either actively or passively. But in doing so, they respond, for instance, to temperature shocks, to chemical stresses, to UV radiations. And you might find that some regulatory interactions or some physical interaction might be turned on and off in a different environmental context, first of all. And then there's another dimension that is very important to take into account, is genetics. Because in the course of our lives, we actually accumulate a lot of mutations in our bodies, and we have different lineages of cells. We're pretty much like mosaics to that extent. And mutations might be very disruptive sometimes, quite often actually. So the network we're studying here might be only partially functional or not functional at all. And to name a few, there are a lot of extra dimensions to that picture. For instance, take the molecular isoforms I've just mentioned, the fact that from one gene you can arrive at many different protein versions. And also the tissue specificity is important, because when tissues differentiate, you have different genetic programs and genes are expressed in a very different manner. So you may not find the same genes expressed in the liver or in neurons, for instance. Right, and then there's the third problem, which is a problem of population average measurements. In most of what we do in traditional biology, you take a bunch of cells, let's say a few millions, you break them, you take the DNA, you measure something about it, and that's great. Let's say you have a cancer biomarker and you say, well, it's really overexpressed in that tumor, let's say in 3 million copies. Well, actually, we don't really know what happens at the population level. We don't know if it's a situation where most of the tumor cells will express the gene in the same level, or whether it's actually quite heterogeneous, but on average it looks the same. And in fact, in biology, it will tend to be this case, because there's a lot of single cell variability. It's actually the topic of my PhD, but I won't go into that detail. So, in a nutshell, I'm really telling you that there's 3 fundamental issues, right? The first one is we need, if we want to push the limits of current biology, we need to take into account time, we need to do measurements genome-wide, we need to also do them across different tissues in different contexts, different individuals, and we also need to do that at a single cell level. So is that actually possible? Well, it might not be possible to do it for all of these aspects at the same time, but any combination thereof is quite useful to have. And so we need to think about 2 aspects. One is, well, it's a bit far, the capacity to acquire such data. Another very, very important aspect is the capacity to handle and analyze very complex data sets. And I really think that for a long time, the answer to those 2 objectives has been no, we can't. But it's starting to become yes now, thanks to a combination of 3 technologies. I'm not going to go into the detail of those very deeply, but the first one is next generation sequencing. You might have heard of it. Something else that I find extremely exciting is called single cell omics. You may not have heard of it because it's very recent. And the third is sort of my field, so computational and systems biology. It's more or less how to handle this data and how to make sense out of it. But I'm not going to talk about it. So next generation sequencing is truly something amazing. It really boomed 5 years ago, I think, or so. And while without going too much into detail of it, I've told you that we had the human genome sequenced already from the 2000s. So we have a sort of graph representative of Jim Watson or John Quirk Venta's genomes, quite specific. And also some people have started to sequence thousands of genomes of different individuals from different ethnical backgrounds and different geographical regions. But roughly this is a reference. And now when you want to use next generation sequencing, what you do is like you extract the whole genome, you fragment it into lots and lots of pieces, you sequence them all in parallel, and with bioinformatics algorithms, you remap it back to individual genomes. And you're still capable of finding variation, like mutations and so on. So you can retrieve that sequence. But the bottom line, really the truly amazing fact, is that it allowed us to go from $2,500 per million bases of DNA down to only 20 cents. So it's something crazy because it's a 10,000-fold decrease. So what can you do with a 10,000-fold decrease? Well, you can sequence a lot more things, right? But also you can sequence them better and in different contexts. Let's say you're a chemist and you realize, there's a lot of proteins that bind on the DNA and they actually have a big regulatory role. So let's, for instance, cross-link all these proteins on the DNA. Then we get rid of the DNA that is not protected by those proteins. And we sequence, with this technology, the DNA that is being captured by the protein. Then you can have a map on the genome of all the events of binding of individual proteins, and you can have a guess about what they're doing in a much deeper level. And there are a whole lot range of derived technologies that I won't have time to go into the detail of, but are really, truly amazing. Or are they? Well, my point is to say that although this is really great, it's only in a population average measurement. But there's a lot of variability. So if you, again, as I said, if you take a cancer, you take a biopsy and you measure something, you will only have a sense of what happens on average, not, you won't capture the granularity, if you will, of the heterogeneity of what happens at the single cell level. But the great news, and I'm going to finish with that, I forgot that aspect, is also that I won't go into detail of this anyway, but this is a really big science, big data project that came out from the NGS. So it's an example of a great application of the NGS. So if you're curious about it, we can discuss that later. But as I said, single cell omics are really like what's taking us to the next step. So it's all based mostly on something called microfluidics. So it's technologies that exploit the fact that you can flow in individual objects into very thin channels and control the speed of the flow, the distance between objects, and where you want to make them move. So it's pretty much like these devices where you have a sort of miniaturized laboratory, and you can make single cell flow in those sort of channels. So you can actually trap cells in individual chambers, lyse them, and make reactions, and then wash the reaction once you're done with it, but you've done a measurement in the meantime. And so those technologies are really, truly amazing, and there's a lot of pioneering studies that show that now everything we could do at the next generation sequencing level can be also done at the single cell level more and more, both for DNA sequencing, but also RNA sequencing, and also at the proteomic level, so trying to study the expression of proteins. And this has amazing applications. And I will just leave you with OneID, which is even further than that, and sort of combines what I've mentioned. Instead of making single cell flow in those microfluidic devices, you can make them flow inside, encapsulate them inside droplets of water that flow into oil. And the interest of that is that droplets are really cool. No, I'm kidding. It's also that you can merge, for instance, you can fuse droplets, you can split them, you can sort them with electrical shocks if they have a certain property. And most interestingly, you can add DNA to it. You can add a droplet with a unique type of DNA that is only found once in one droplet. You fuse it to a single cell. And what you've done here is that you've barcoded the single cell. So you can make a measurement, and then you're able to sequence the DNA and say, well, I can identify that particular cell uniquely. And the interest of that is also because the droplets are stable. You can keep them in your fridge. You can redo experiments, and it lowers down the cost like crazy because you only need very, very small volumes of chemical reactants to perform a lot of the reactions you need. So it means that if those technologies become broad and commercialized and so on, it will have a huge impact on our capacity to understand the three fundamental aspects I've mentioned, especially time, multi-dimensions, and single cell level, right? So I will leave you with that, and thank you for your attention.
Thank you very much, Guilhem. Any questions? We have time for at least one, maybe two. This is just a question of sort of general curiosity, but the single cell omics, like the fluidics techniques, are they designed mostly for like human-sized cells? Because there's, my understanding, quite a lot of variation in cell size. So if you're basing it on fluids, could you use it on something like a prokaryote or like an amoeba?
Yes,
okay.
Yes, it is possible. So of course it means that the inconvenience is like you need different type of devices, basically that are adapted, for instance, yeast cells, if you're still talking about eukaryotes, and some for bacterial cells as well design. But that's not really something I'm working on, but I've attended some conferences on that topic. And indeed, it's possible. It's just a problem that you need to change the device.
Okay, yeah, we have one more question.
This is not a science question at all, but I wonder if you've thought about the ways in which some of the barriers to studying things like the way that these things happen in time, or studying like heterogeneity, you know, rather than averages, are not just technical difficulties, but also conceptual difficulties.
Right.
Because these are things that we see in my field, which is musicology, where people have only recently started to think about the way we experience music in time, rather than looking at text. And so I'm wondering if you've thought about sort of relationships with other disciplines.
No, this is a great question. Thanks for that. I think, in a sense, I've tried to convey that message that it's important to try to characterize what are the fundamental limitations we have. And I think it's a sort of, you know, top-down, like bottom-up approach. Like you can really go to the facts and then try to see if they work, you know, experimentally, empirically. So to answer your question, I think it is true to a large extent that people get a bit trigger happy, you know, with publications, and they will say, well, we solved that problem at a genome scale level. And then you discover, you scratch a bit and say, well, no, you really missed a dimension that is truly important. But it's more a matter of communication. I think scientists are quite aware that, you know, all these dimensions are truly important, but they're just extremely difficult to challenge and to use if you don't have that data set. And also what I forgot to mention is, you know, even if we acquire those data, we really need to have bright people to analyze them. And that's not something trivial at all, but it's progressing over time, and thanks to a lot of different disciplines, in particular IT, like business intelligence as well. So machine learning, for instance, is truly important for bioinformatics and for this sort of field as well.
Thank you, Guilhem.
Yeah,
Thank you very much.